Hello there, and welcome to my lecture on chemical digestion. This is one of 4 I'm asking you guys to check out while I'm away in Portland. I'm doing a presentation. Hopefully you haven't minded this make do of recording lectures and as best I can with my limited skills in recording. Anyway, so we're going to be talking about chemical digestion, filling out the nutrient table that I passed out to you guys before we left. And hopefully, sorry about that. We're, this is actually my last one that I'm recording, but maybe it's not the last one that you watched. I do expect it to be on the long side. Okay, so we have to talk about chemical digestion of carbohydrates, proteins, lipids, and nucleic acids, and we need to fill out the nutrient table. So, because I don't really have a board, I'm going to just do the best I can. Mary. So, we're going to start with me just talking. I'm not going to write anything down yet, just going to give you guys an overview of, let's see, not all of this stuff. I hope I'm going the right direction. The digestive enzymes. So we saw in saliva the salivary amylase made by the salivary glands. We saw lingual lipase for lipids being released by the tongue. So not in saliva, but being released by the tongue into the mouth to work on fats. However, remember that lingual lipase didn't get activated until it hit the acid in your stomach. In the stomach, we saw pepsin working on proteins, and we saw gastric lipase also being released to work on lipids. So right now we have carbohydrate digestion beginning in the mouth, protein digestion beginning in the stomach, and lipid digestion beginning in the stomach. Pancreas makes all enzymes for all foodstuffs, right? Remember, the pancreas is an endocrine organ because it releases hormones, insulin and glucagon into the blood to manage blood glucose levels, and then it's an exocrine gland because it releases every single type of digestive enzyme into the pancreatic duct, into the duodenum. So there's amylase for carbohydrates, there's trypsin for proteins, chymotrypsin, elastase, other sorts of peptidases, and then there's pancreatic lipase for lipids. Excuse me. The pancreas also is the only structure, well, other than brush border enzymes, I guess, is the main organ for producing nucleases for digesting DNA and RNA. Find that, in products that you eat. And then you have these interesting little enzymes coming off of the brush border, the microvilli in the villus. And we have brush border enzymes for carbohydrates, like sucrose, lactose, maltose. We have them for proteins, but that's activating trypsin. Okay, here we have amino brush border enzymes for breaking down proteins, and brush border for breaking down nucleotides. Oh, but no brush border for lipids. I need to remember that. All right, well, that's good to know. This is an overview, but we're going to talk about this. So, guys, I don't want you to get too I'm going to go look at this picture quickly, but we're going to dissect this when we fill out the table together. So what I have is carbs being converted into simple sugars that goes across the small intestine, microvilli, and enters into a blood capillary. We have proteins being chemically digested via enzymes to amino acids and smaller polypeptides, also going across the villi and into a blood capillary. And where is my I don't see my RNA and DNA, my nucleic acids, but those are also going to go into a blood capillary. Then what we have are my lipids, and lipids are crazy. And we're going to look at lipids in more detail, but lipids, right, they don't like water. So we need to package them and make them like water. So lipids are packaged a couple of different ways, and ultimately they don't go into the blood capillary, they go into that lymphatic lacteal. Oof, well, whatever. Great handwriting. Into my lymphatic system, and ultimately then get dumped back into the blood supply where the lymphatics made it at the subclavian veins. So let's look a little more closely into some of these digestions. So, what I have is carbohydrate digestion. Oh, why is this? Oops. Oh, okay. Sure, keep them. I want to kind of toggle back and forth. So we have carbohydrate digestion. We'll make this work, where it's starch and, other saccharides. Oh, interesting. And they get broken down into simple sugars. The enzymes we saw for breaking down carbohydrates is salivary amylase in the mouth, pancreatic amylase working into the small intestine, coming off of that, and then brush border enzymes, all ending in ASE, that are doing it in the small intestine. From there, they're getting absorbed into a blood capillary in the villi. So let's go back to this guy here. All right, so carbohydrates. Oh, I wonder, am I in drawing mode? But I'm not. Let's go into drawing mode. Let's do blue, right? And let's make it be, okay, that looks good. Alrighty, well, let's, okay, so carbohydrate, my subunit is a simple sugar. Oh no. This isn't gonna work, is it? Haha. Nope. Okay. Why is that not working? Stinker. Okay. I guess maybe I'll try typing it. They get broken down into simple sugars, like glucose, fructose. Oops, how about I spell it right? And one enzyme I saw was salivary amylase. The salivary glands make it. This works in the mouth. We had pancreatic amylase coming off the pancreas. It works in the small intestine. But then we actually also had, no, I just got to go with it. Brush border amylase coming off of the small intestine brush border, microvilli. Also working in the small intestine. All this gets absorbed in a blood capillary. All right, so what I really want to point out is that this begins in the mouth and ends in the small intestine. Ends in the small intestine. All right. So now let's go back to this guy. Maybe I'll keep it big. Okay, so, and for some reason I did it again. Oh, is that, protein. That's right, I screwed this up and didn't pull in the protein one. Alrighty. So proteins, let's just go with this guy. Proteins get broken down into amino acids. Might have already had some, oh, that's right, I'm not writing it, I'm typing. Proteins get broken down into amino acids. And this enzyme, the first enzyme we saw for that was pepsin. That is made in the stomach chief cells. And it works in the stomach. And then we saw pancreatic. Well, it's called trypsin, right? Made by the pancreas. A location in the small intestine. Now, when I am pepsin or trypsin, and I chemically digest proteins, I break proteins down into their subunit, I would conceivably digest a cell, so it has to be released in an inactive form. Inactive form pepsinogen that is activated in the acid of the stomach. And the same for trypsin. This is released in inactive form, trypsinogen. So I do not digest the cells that make me. And of course, we also will have the brush border, peptidases. Peptidases made by the small intestine, brush border microvilli. All of this is in the small intestine, and amino acids also get absorbed in blood capillary. Okay, so let's see where we're up to. All right, so let's go back to this guy. Text. Okay, nucleic acids there. Right, digesting DNA and RNA, and I have the ribonucleases and the deoxyribonucleases coming off of the pancreas and the small intestine and the brush border. And these are also going to the blood capillary. Oh, good point. I like that it then says then to the, then to the liver via, so let's type that in, blood capillary, then exit small intestine air mesentery via hepatic portal vein to the liver. Might as well be fancy, right? Yeah, that's cool. Same for this guy. Now let's get, oh dude, lipids. Let's do nucleic acids. So nucleic acids get broken down into nucleotides, which are a nitrogenous base, phosphate, and sugar, either ribose or deoxyribose. Okay, so we have pancreatic nuclease, nuclease for RNA, pancreatic deoxyribonuclease. Would that be any longer? Nuclease for DNA, and then brush border. Brush border nucleosides, lipases from either the pancreas, right, or the microvilli, brush border. So nucleic acid chemical digestion begins and ends in the small intestine. And this is also, do we still have that whole shebang? Gets absorbed in the blood capillary, exits to the liver via the hepatic portal system. So now we come to lipids. So let's look at the PowerPoint first. I checked to see if I was recording because I'm like, oh my gosh, if I forgot to hit record and I'm this far along, I would cry. I'm recording, thank goodness. All right, so bile. What is going on with lipids? So lipids, right, are hydrophobic. They don't like water, and they're hard to digest. So first I have these bile salts, and these bile salts take a big fat and they make it small fat, right? So I'm now little small fat droplets, which means I have increased surface area for enzyme activity. So the bile salts break it down and they kind of cover it all. They coat it to keep it in water. So when I am this bile salt, small fat droplet, like entity, fat droplets with that. Let's see right here. What they're called these micelles. So these micelles are bile salts with smaller fat droplets, and this is what is able to cross into the villi cells. So they cross into the villi cells, and then they're repackaged with proteins, and now they're called chylomicrons. And the chylomicron then leaves, and this big bulky molecule can't enter into a blood capillary, so it enters into a lacteal, which is cut off. I don't think, because I'm recording, can I grab that a little bit? Okay. So now you can see that these chylomicrons enter into enter into lacteals, where they are carried in the lymph, and then ultimately they end up in the blood supply. So these get to the liver via the hepatic artery, right? Because they're going in at your subclavian vein, and then it would be into the hepatic artery to the liver for processing. So let's write that. Sure, keep it, whatever. I know we're getting there. So let's go back to this. Okay, so lipids. Lipids get broken down into fatty acids, glycerol, and kind of eventually will exist for travel as a triglyceride. So glycerol with 3 fatty acid tails. Okay, so we saw lingual lipase coming off of the tongue. That working in the stomach needs acid. Environment. I know, my stupid phone. We saw gastric lipase coming off of the chief cells of the gastric pit. That is working in the stomach. And then we also saw pancreatic lipase. And pancreatic lipase is going to do 80% of the lipid digestion, lipid chemical digestion. This is coming off of the pancreas. Oh my goodness, Lord, I'm not a fan of where it went. And this activity, of course, will be in the small intestine. So now let's just make sure we know what the heck when it comes to absorption. Where's my pen? Hello. I'm going to go with black, and I guess we're going to make it like that. Okay, so lipid absorption. Of course, it's on the last one, and I'm finally writing better. Absorption. Ugh. Okay, so first, the bile salts plus small fats are packaged as micelles. Oof. You saw that? I'll try to write it better. I'm sure everyone's complaining about my handwriting. Micelles. Close enough. This then will enter into villi cell. I'm really not writing this bad. Where it will then get broken apart and repackaged. So the interesting thing is that, well, here, let me repackage it as a chylomicron, which is the fats broken down into triglycerides and proteins to keep it soluble. The bile salts get recycled They're then pulled off and recycled. Some will leave, but a lot of it gets back in and made back into bile, but then I can excrete some excess cholesterol, excreted with the feces, right? Oh boy. All right, so this is kind of a little bit aside of the story of bile. So these chylomicrons then will leave the villi cell. Now this is actually the letter A. I know, it looks like it's L-E-A-V-E. The villi cells, chylomicrons will leave that and they will enter into lymphatic, lacteal. This lymphatic, these chylomicrons then will get to the blood supply, but I kind of like what I added to say that they will eventually get to the liver via the hepatic artery. That's kind of cool. I can never ask you that, but I still think it's a nice little connection of what's going on with how to get to the liver for processing, right? So lipids, they are so difficult. Okay, how are we doing for time? I have no idea. So let's get back to PowerPoint mode. Oh, we're getting there. And what do I have left? Oh, I think we pretty much have talked about all of this. This is what we pretty much talked about as all of my subunits of amino acids, monosaccharides, maybe even some teeny weeny fatty acids. Okay, those can get into the blood and go to the hepatic portal vein, to the liver for processing. And then we have the triglycerides with proteins as chylomicrons and a lacteal, right? That's this green guy. And this travels through all your lymph nodes, enters into the subclavian vein, to the heart, and then would get to the liver via the hepatic portal, the hepatic artery. Okay, I like this picture. So this is the in, and this is like what we use of it. So we make, I just love this, 1 liter of saliva. This is per day. This is one of my favorite slides. And we're there, my favorite slide. 1 liter of saliva. Maybe you're drinking 2.3 liters of liquids. Your stomach makes 2 liters of gastric juice. You make 1 liter of bile, 2 liters of pancreatic juice, intestinal juice, another liter. So everything that's kind of your input, 9.3 liters. Now look at 8.3 liters is absorbed. And in my large intestine, I absorb another 0.9 liters of mostly water. Right, so total absorbed is 9.2 out of 9.3. And thank you, I will keep 0.1 liter in my feces so that things move easily. If I were to absorb that 0.1 liter, and then golf balls are going to be coming out of me, and that's not so fun. That just blows me away. 9.3 in, 9.2 kept in, and 0.1 released, right? Oh, and that's it. Hold on, I'm excited here. That is the chemical digestion and absorption that happens in our body. And our last, well, for me, the last one, hopefully 1 of 4 for you, and hopefully you learned something through this recording. All right, I will see you guys on Wednesday, so take care. I'll be thinking about you. Yeah, I'll be thinking about you a little bit. I'll be honest. I'll be busy, but it'll be good. All right, take care. Bye.